to you. So, Professor Qian Kong has majored in biological sciences at Peking University, where she learned to predict structural and functional properties of proteins from their sequences. After that, she got her PhD from University of Texas Southwestern Medical Center, and after graduating, she was a senior research fellow at the University of Washington at David Becker's group, where she set out to model protein-protein interactions on a proteome scale, a direction combining her expertise in large-scale data and protein evolution. This approach turned out to be very powerful, showing comparable performance to large-scale experimental protein-protein interaction screenings. Since 2020, she has started her own group, and she is an assistant professor at University of Texas Southwestern Medical Center, where she continues her journey in computational biology at large scales. So with further introductions, it's my pleasure to give the stage to Yuki.
Oh, so should I start it now?
Yes.
Yes, please.
Yeah. Thanks for the nice introduction. So I'm sorry about the late announcement. So I didn't reply to email in time. But thanks for the people who still come, and I hope that you will enjoy the talk. So today I will talk about is computing the human intactome. This is some project I wanted to do for probably 10 years, and we finally managed to do it in the last year. So this is just a general background of what we are doing. So this is now a very well-known phenomenon called coevolution. So what is coevolution? Like if you study sequence alignment very hard, then you probably will see residues that seem to be evolving in an interdependent fashion. So these like correlated changes in sequence alignment usually correspond. It's called coevolution, and residues that are co-evolving in most cases correspond to the residues that will come close to each other in the 3D space. So then you can use this coevolution phenomena to help predicting structures. So, but the methods to analyze coevolution have been evolving over the last decades. So like in like more than 20 years ago, so there's not too advanced techniques, and usually for studying coevolution, people look at one pair of residue in a protein at a time. So just like what I was showing you just now, like by like analyzing one sequence alignment, identifying what residues are co-evolving. So these methods did reveal some interesting biological insights, but they are not powerful enough to be useful for structure or interaction prediction. So then about like a decades ago, then people invented this so-called global statistical method. So what are global statistical methods? So instead of looking at one pair of residues at a time, global statistics allow people to look at all pair of residues in a protein at a time. So then by doing that, they were able to understand which pair of residues they are directly like co-evolving, which ones seem to be co-evolving due to transitivity of this coevolution. So this method yield a small breakthrough within the structure prediction field, where people can just, starting from the sequence alignment, predict what residues will come close to each other in the 3D space. And that will help people to be able to predict the topology of a protein from scratch. So it was a very exciting improvement. That's when I got interested in this field, but it did not really reach to people outside our field. But then finally, due to AlphaFold, and there's also Rosetta Fold, and all these deep learning methods, they again revolutionized our way of looking at coevolution. So because these deep learning methods learned from the entire protein universe and try to correlate sequence and structure, so they are the best tool to infer this coevolutionary signal and using it to for structure prediction. So then now everybody knows about it, and then. But every like a huge improvement has a long history. So I just want everybody to like appreciate this long history before AlphaFold happened. So then with all these methods to understand the coevolution and predict structure, then obviously you can apply it to single protein. Then starting from multiple sequence alignment, you get coevolution, and you can now accurately predict structure of nearly any single protein. And obviously, you can also extend it to use this type of method to predict interaction between proteins. Because if two proteins interact, then whatever residues on the interface are also supposed to co-evolve. So all the methods developed for single protein can be used for protein complex. Although, like based on our experience in the most recent CASP, it's true that for almost every single protein you can correctly predict its homology, but it's not yet true that people always can get the protein complexes right, even with the most latest development. So then I will just briefly introduce to you like how would we predict whether a pair of proteins interact. So let's say you have two proteins A and B from human, and then you want to predict their interaction or whether how they interact or whether they would interact. Then what you would do is to gather like homologs, actually orthologs for these two proteins from all different organisms. And then if you assume protein A and B work together and bond to each other, then you would assume that they would evolve together. Then you can make an artificial like a concatenated sequences, like fusing these two sequences together and see, then you can start to analyze if there are co-evolving residues between them using all the methods that are established for single proteins. So, and then after identify these co-evolving residues, then you could try to dock to these two proteins and see if the co-evolving residues you identified do correspond to possible contacts on the protein-protein interface. So this is like the old school way when I was doing postdoc. This is exactly how I was doing it. But now, of course, a lot of this are packaged inside the black box of AI. But we believe that what AI learns are similar things. So they are like co-evolution. It learned to do co-evolution and also learned to model structure. So it learned better. It can like do these things in a more coherent fashion and achieve much better performance than the traditional statistical method. So to extend this kind of analysis to a protein-wide screen, then you simply need to repeat it for 100 millions of protein pairs. So it is as easy as that, but it's pretty challenging to really extend it to be a protein-wide PPS screen. So the first very important challenge, it is computationally very expensive. Let's say my goal was to really do it for human. The human will have 20,000 proteins that correspond to 200 million protein pairs. Like I have a small academic lab. This is really very expensive to do. And another very important thing is like if you are doing this PPS screen, you are facing an extremely low signal to noise ratio. Let's again take a human for example. So although you have 200 million protein pairs, it is estimated that the true interactions in the human proteome is only like 74,000 to 200,000. So this will correspond to a signal to noise ratio of 1 to 3,000 to 1 to 1,000. That means for every 1,000 possible random pairs, you are trying to predict only the one pair that is interacting. So then when these things, so with both these considerations, then you want to constantly balance between the speed of your screen and the accuracy of your screen. So then over the years, our like trade-off between these two factors is we would usually use a hierarchical pipeline. So we will start with some fast but not too accurate tool and apply it to many millions of protein pairs. And then from those millions of protein pairs, we would identify the candidate interactions that have higher chance of interacting, and then we only apply those slower but more accurate tool on those candidate interactions. So like we balance between the cost and how much we can gain at the end. So, but the tools that we put to this pipeline is keep updating as the field progress very rapidly. So in the beginning, all these I was only using these statistical methods, and now every pipeline that we use is heavily relying on AI, which is now to be the best method for inferring co-evolution and identify interactions. So then before like I started with human, I actually started with bacteria. So that was before any of these AI tools were available. So we were able to make a small number of predictions in bacteria. That was the work I did. I did when I was a postdoc in the Baker lab. So then when I was in the Baker lab, we did start to thinking about doing this for eukaryotes, but it never worked because without those artificial intelligence methods, the statistical methods are just not powerful enough to extract signal from the shallower sequence alignment for eukaryotes. But as soon as I started my own lab, then there's the AlphaFold got to like shock the field. Then we immediately start to go back to this problem and thinking, okay, maybe now with this better tool, we were able to solve this old problem that we were thinking about for a couple of years. So then we just decide to go back to the east. And so we work on east because east has a relatively smaller number of proteins. So it has like about 20 million possible protein pairs. And the yeast is good for any PPI studies because it is established model organism for this. So because there's like yeast to hybrid, so that is just like all the experimental data for yeast, it seems to be a little more reliable than done on any other organisms. And then you can easily put together like a positive control, which consists of those bona fide interactions validated in multiple experiments. And then you can also put together a negative control, which consists of pairs that has no previous evidence of interaction. So it is important to note, like since the start of my journey in this direction, I was always trying to put together positive control versus negative control with a signal to noise ratio on the order of 1 to 1,000. Because my ultimate goal is to use this method to identify like true biological interactions. And we do really go deeper to study these interactions and we help collaborators. And we don't really want to like make prediction and there's a lot of false positives. So keeping this very real and very challenging signal to noise ratio allow us to allow our tool to be really useful when you apply it to real case scenario where you are indeed facing a 1 to 1,000 signal to noise ratio or even worse than that. So, and then another thing that I need to mention that is probably not too important for this audience is like we need to constantly monitor our performance as we go through our PPS screening pipeline. And we are using precisions and recalls to monitor the performance. So just a reminder of how precision recall work. So for example, you have a set of predicted interactions and you also have a set of true interactions. And you would measure their overlap. The larger their overlap, the better your method is. So then to quantify like how large is the overlap, you can use this overlap to divide by the number of predictions. That you get a so-called precision. That is like how clean, how accurate your predictions are. And meanwhile, you can also use this interaction to divide by the size of the true PPIs. So that will indicate how complete your predictions are. So then these two features, they're usually reversely correlated. So it's hard to get very well on both. Just to help you to build some intuition on how these numbers usually look. So what we usually monitor is this so-called precision versus recall curve. So this is like as you try to predict more and more interaction, then you monitor like how clean your predictions are, and meanwhile how many true interactions you are able to recover. Then using the positive and negative control data set mentioned in the previous slides, then you were able to like try to make a precision recall in the curves like this. So if you have a totally random predictor, then you are just ranking all those proteins like randomly. Then you will get a precision and versus recall curve that is very close to zero because you have 1 to 1,000 signal to noise ratio. So like every one positive, you will get 1,000 negative. So you will get a precision that is close to zero and from the beginning to the end. And let's say if you have ideal way of predicting, then you will get all your true positives and you will rank them higher than any of the negatives. Then you will get a precision versus recall curve like this. And then realistically, you usually get a curve in the middle. And the area under this curve indicates how powerful your screening method is. So then this so this precision versus recall curve is important because as we run our screen, this is our primary measurement of our tools. So then this shows like why we were not able to do it using the statistical method. So let's say if we do apply statistical methods to this East PPF screening task, this is the precision recall curve you will get. Well, it's not like too terrible. You probably can predict about like a 1% of all the interactions, but that's kind of meaningless. You are probably going to predict like interactions between ribosomal proteins. So then as soon as this AlphaFold came out before they released their code. So I've been like again collaborating again with my postdoc advisor working on building like a version of Rosetta Fold inspired by the AlphaFold ideas. And more importantly, so I'm interested in like making this Rosetta Fold much faster so that we can do it for protein-wide PPS screen. So and a lot of these benchmarks were done by like my research scientist Jimin. So then as soon as we made this like a Rosetta Fold and a lighter version of Rosetta Fold, which is like 20 times faster than AlphaFold, so it's feasible for protein-wide PPS screen. We can see even this faster tool showed a lot. That's very significant improvement over the global statistical methods. So that's good. Like but then we probably can use this like instead of the statistical method, use it as a like a first step screening method. So then we can apply Rosetta Fold to millions of yeast protein pairs and identify a smaller subset of protein pairs that are more likely to interact. So indeed, we use that and we identified about over 5,000 like quite likely and quite possible protein-protein interactions. And then we just applied AlphaFold on these 5,000 like candidate interactions. Then we see some amazing thing like, so if we apply AlphaFold on top of this selection by Rosetta Fold, we see like AlphaFold can perfectly rank the positive and negative. So it was able to achieve extremely high precision, although the recall is limited by like our initial selection of these 5,000 proteins. But you can perfectly distinguish the positive from negative within this site. So then this precision versus recall curve on the right shows like we were able to identify about 28% of the true interactions at a precision of 95%. So that's important because if you want to make a biological discovery, you want to make sure that your predicted site is totally dominated by true interactions. So then we were able to achieve that, and we were very happy about that. So I'd like to mention that this very ideal performance is not just achieved by AlphaFold. So later on, we also did the benchmark. If we only use AlphaFold, can we reach like this like a perfect precision recall curve? We cannot. So this Rosetta Fold step was indeed also important. Although AlphaFold give us an advantage, like because AlphaFold can also give you a very accurate 3D structure. So then we just applied this pipeline to yeast, and then we were able to predict 1,500 interactions. So and the yeast is so well studied, so about half of our prediction, their interaction is known, and the 3D structure is also solved. But also about half their interaction was sort of supported by previous experimental evidence, but how they would form a protein complex wasn't known. And there's a small fraction of novel interactions. And since that was like when AlphaFold was just released. And it is also even for these, like their interaction, just to model their 3D structure, give you a lot of biological insights. And some of these protein complexes are have very important function. But knowing their structure, we were able to make a lot of interesting observations and gain biological insights. I'm not going to go into too much detail of each complex, but each is associated with a biological story. I will just show one example. Let me see. So this example is a protein complex we were able to identify and model using this pipeline. It is a GPI transmedase. So the function of this complex is just to attach GPI anchors to proteins. So the components in this complex was more or less known, but how they would interact each other, which pairs would directly interact, and how they would form a complex wasn't known. So we were able to model the model like a complex of this enzymatic this large enzyme using AlphaFold, and then using this model we were also able to identify the catalytic site and a channel for potential substrate to go through. So we released this prediction in 2021. Then in 2022 people solved this complex in human. Then you can see that now it's no longer surprising that indeed these modern AI network they can predict structures quite accurately, and then they were able to produce the biological insights that before AlphaFold was only possible if you solve the structure. So and then it's time to really move on to human. So then before moving to human, then we were wondering it's like human, is it better studied than yeast? Like human is so important, maybe it's all so well studied. But the answer is good for us that the answer is not true. So human intactome is not well studied at all. So let's just go back to some statistics. So we mentioned that for human, we have 200 million protein pairs, and the estimated number of true interactions is on the order of like 74,000 to 200,000. But however, interested, so if you just dig into PPI databases and just find which pair of human proteins had some experimental evidence for their interaction, you will easily find more than 1 million. So then what does that mean? So like you find more than 1 million pair, but the true interaction is like much smaller than that. So that means like all these databases, they were like plugged by potential false positives in experimental screens. Especially if you pull together different experimental screens, then you really get like all the noise from different experimental data. And why like a PPI experimental study is noisy because proteins are sticky, especially you put them in non-physiological condition. So for example, if you do like crystallographer, then any protein will form a crystal. Well, not any, many protein will form a crystal, although they don't really self-associate in the physiological condition. So this protein has just this sticky nature that making an experimental study with PPI also very challenging. And this experimental data set is rich in noise. So then on top of that, then how many pairs have experimental structure? So up to now, it is about like 10,000. So that means so the PPI data, experimental data is noisy, and those really, really very short interaction is only a small fraction of that. So another analysis that highlight the challenge of identify true interaction from human is the analysis based on different PPI databases. So we gathered like BioGrid string physical interaction from string and UniProt. These are well-established databases for protein-protein interaction. If you try to identify the confident PPIs from each data set and try to see like how well like they overlap with each other. Then you will see so we take like over 60,000 confident interaction from 3 databases and we see their overlap is just 4,000 pairs. So that means like it's really hard to judge like from these experimental data what are the real interactions happening in human. So before we started our human journey, because one easy thing we were thinking about doing is like maybe we can do what we do in yeast in human, but actually Annie did that and better than what we can do. So then we decided, oh, then what can we do that is still meaningful contribution to the field? So we decided to go back and using our expertise in large scale data and AI method development, try to improve this pipeline and see if we can push the boundary further. So so in our initial trial of trying to improve our method for PPI identification, we made a newer version of Rosetta Fold, like faster version of Rosetta Fold, just by incorporating the insights we get from the AlphaFold after its release, then we can learn better from there too. So that did improve us, it did introduce a small improvement, but we were not happy enough about it because if you really do this huge, very costly screen and end up only identifying 10% of true interactions, that just doesn't sound like worth the money. But yet, due to our East paper, people in my institute is assuming that I'm the person who will be able to do it. But realistically, if they give me if 10 people approach me, I probably will be able to help one of them. So this put me into some imposter syndrome. So I was like, oh my God, what am I going to do? Like this, everybody expecting me to do it, and I just cannot do it well. So then I was this my imposter syndrome was solved by my brilliant postdoc who is really dedicated to make this happen. So then you can see like so this is where we started with like this is the performance of our East tool. Then through a number of very like a heroic improvement she made, we were able to improve our method by maybe fivefold. This is really like rare to see like in a bioinformatics you can have like fivefold improvement. But I'd like to note like this huge improvement is also due to that this task was so challenging and we started with a tool that is really not adequate. So we were able to make a huge improvement. Then I will next introduce you how she did it. So it's mostly relying on better AI tool and a large amount of data. So then to make our tool better, our first hypothesis is maybe the small size of a training data set is limiting the performance of current deep learning network, including RussellFold and AlphaFold. So for this network to be able to predict the protein-protein interactions, it's a common practice to use the PPIs from PDB to train these networks. However, although there's a lot of like PDBs that have protein-protein interaction, but after removing those that are showing homology to human pairs because we don't want any data leakage and clustering to remove the redundancy, then you really only will have like little more than 10,000 clusters of diverse PPIs. So this is not too large a training data set, but where can you get a larger data set? So we decided to look into AlphaFold protein structure database. Alpha AFDB doesn't have it only consists of protein monomers, but inside protein monomers, they are domains, and domains they are evolutionary units that got recombined to create these proteins, and these domains they do interact. And our hypothesis is that maybe the interaction between these domains will resemble protein-protein interactions. Then we can use the domain-domain interactions as a training data set for PPI prediction tool. So luckily, like before we dig into this PPI, we go back to this PPI task. We also developed a method to analyze AlphaFold models, in particular to identify evolutionary units from these models and try to classify them. So then this kind of tool allowed us to really identify domains from each AlphaFold models, and then we can identify domains that come close to each other from a confident interaction, and using that as a to enlarge our training data set. So these are the features we use to pass domain. So we take the domains annotated in interpro, which is sequence-based annotation. Then we add the structure features from AlphaFold model, including like inter-residue distances and predicted alignment error. And this allow us to predict domains. For example, in this protein, we pass into 3 domains, and you can see each pair of domains seem to be interacting with each other. So then going to this domain-domain interactions in AFDB allow us to significantly enlarge the training data set. So we were able to make a training data set that is about 20-fold larger than what is traditionally used in the field. So here I'm just showing you a couple of protein-protein interactions that we found. It's a random selection. Then you can see that they do kind of resemble protein-protein interaction interfaces. So then after we get a better training data set, we decide to also optimize the Rosetta Fold architecture for PPI prediction. So we tested a number of different modifications, and to our surprise, like so the modification that made the most significant change was when we removed the 3D structure feature from RussellFold. So that was surprising because if you compare like RussellFold and AlphaFold, the major difference is how we treat the 3D features. So in AlphaFold, you have evil former that integrate sequence alignment features and pairwise features that kind of encode this coevolution and residue contacts. And they also have a separate structure module that help to take these residue interactions you derived from these 2 features and try to fold them into 3D structure. In Rosetta Fold, we made this modification where we directly allow the 3D features to talk more frequently to the MSA features and the pairwise feature. This change was important for protein design because this more close association allow you to easily go from a 3D structure to a sequence. But it probably wasn't the best option but best option for our prediction. It turned out if we remove these 3D features, just only using MSA and pairwise, we were able to get a huge improvement. So that also now we can be smart and think about why didn't AlphaFold choose to put the 3D features in the evoformer. Maybe they also observed something similar. If they do put it in, they probably also get worse performance for predictions. So then we just put together this new optimized architecture and along with our training data set. It's worth mentioning we also have, in addition to true interaction, we also put some negative interactions with pairs that shouldn't interact to allow the network to learn both sides. And then we made a much shallower network again, so to be a lot faster than AlphaFold, and then we trained the new model. So then another hypothesis is maybe the shallow sequence alignment for eukaryotes is limiting the coevolutionary signal. This is particularly true for human because many sequences in human they are specific for higher eukaryotes. You simply don't really have a very deep and diverse alignment for human specific proteins for higher eukaryotic specific proteins. So this is contributed by the fact that a lot of the sequenced eukaryotic genomes in the databases are not annotated. Like if you take an NCBI and you get all their genomes, and only 20% of the genomes are associated with the predicted protein sequences. So although the genomic field did a lot of sequencing, but they are not really used by the protein science field because we don't even see the proteins from them. So, and even more, like in addition to the draft genomes that are non-annotated, there's also a lot of genomic data sets that in this SRA database from NCBI where people deposited their genomic sequencing, but they did not really assemble these genomic reads into genomes and they did not predict proteins from them. So there are like 30 petabytes of data in this database, just awaiting for people to dig into it and make discoveries. So actually, so just like in addition to my training in the protein science field, actually I spent 4 years in the genomic field when I was a grad student. So and like a lot of the genomes, well, not a lot, well, but at least maybe a couple hundred genomes in NCBI was actually deposited by people like me, by me, by our team. So then we also will frequently deposit our genome, but we don't always deposit the protein sequences we predicted from them because to deposit that, then you need to do a lot of work. You almost need to hire a dedicated data depositor. So because there's a lot of rules in NCBS, it's difficult. So that's why people don't do it. We also didn't do it. So, but due to this genomic experience, and also my postdoc we also used to work on genomics, we were able to build very efficient pipelines to mine all these genomic data set to predict protein sequences from them. So we just took this very large amount of data and try to get useful information for us. So and how did we do it? Just very briefly, we started from the genomes with proteins, and then we try to use these annotated genomes to predict more proteins from the draft genomes without annotations. And then we have more predicted protein sequences, and we use this to guide us to directly assemble protein sequences from genomic reads for higher eukaryotes. And a lot of very excellent and fast tools developed by people in the field was very helpful for us. So then, as a result of this data mining, we were able to get like more than 20,000 eukaryotic species. We can actually get more because for this genomic data set, we only focus on the chordata and insects. But you indeed, they are even more diversity than this. But this was sufficient for us to make a huge improvement. So by using this data set, we were able to get sequence alignment that is about 7-fold deeper than what people can get by just using sequences in UniRef. So then does this deeper sequence alignment make a difference? Yes, it does. So again, we assemble the positive and negative control set that's 1,000 times larger and try to benchmark how much our improved sequence alignment is going to help. By the way, we call our sequence alignment omic MSA because it's directly assembled from genomic data. Then we can see using omic MSA, we were able to improve every method that we used for our PPI screening. So that's good. So then then overall, like let's put all these together. So we have omic MSA, then we have a newer version of Rosetta 4 that's the tailored for PPI prediction, and we were able to improve significantly over over our previous tool. So this is like combining both omic MSA and our new tool. So this is the precision versus recall curve you can get. So it is better than if you just use collab fold. And this better is important because our tool is 20 times faster. But nevertheless, like our omic MSA can improve Rosetta fold, it also can improve AlphaFold. Let's say if we put omic MSA together with AlphaFold, then we make AlphaFold even better than the Rosetta fold. So, but anyway, then if you compare the time, then you can see that our Rosetta fold is a lot faster than AlphaFold, uh, and that allow us to do a protein-wide PPS screen. So then after we submitted our paper, the reviewers start to ask the questions. A lot of questions helped us to really dig deeper into this problem and gain more insights. So one question predictor asked is, you see, we were always comparing our tool against the AlphaFold. Then we saw, wow, AlphaFold is the gold standard, like that's the best tool that you should compare to. But reviewers don't think that way. Reviewers think, well, AlphaFold is not really designed for PPI prediction. You should compare to tools that develop for PPI prediction. So then, yes, indeed, then we gathered a bunch of these new deep learning methods, most of them based on protein language model for PPI prediction and see. Well, many of them, they just had a terrible prediction, like especially when you apply, because we always apply benchmark our methods using a 1 to 1000 signal to noise ratio. And many of these tools, they were trained based on like 1 to 1 or 1 to 10 signal to noise ratio. When you apply them to real world tasks, it simply doesn't work at all. But you do see there's one method that seems to be having a decent performance. Then we go into their training data set, we see they exactly use the human PPI to training. So a lot of the cases that in our benchmark set was exactly in their training data set. Then we try to remove this data leakage, but not even too extensively. We can already see, well, yeah, the performance really dropped. So from this experience, what did we see? Like a lot of these network dedicated for PPI prediction, they they are working by memorizing the training data set. If you don't allow it to memorize, then its performance drop drastically. So that so I think there's some general like difference in how we do this PPI prediction and how the machine learning people do PPI prediction using large language model. Because usually what they do is they take the ESM embedding and just convert the ESM embedding to a PPI prediction tool. Then the problem is they usually train using using the fact whether two proteins intact. So then they can only have like about like 100,000 training data points. Whereas for us, we are training this on the residue level. That each of our training case is giving you many possible contexts. So each protein pair will give you like 100 to 1,000 residue contexts. So our effective training data set is like so much larger than if you just consider the fact two protein intact. So doing it on the residue level give us the advantage of fully utilizing the data set and making very large, making relatively much larger training data set. And also we don't directly train on this PPI prediction test, and yet when we try to apply it to a real PPI screen, we were able to face this 1 to 1000 signal to noise ratio, whereas many other like protein language model based tool, they were not really trained with this realistic goal. So then another commonly asked question is how do these things work in transit interaction? So we identified the likely transit interaction as like a protein-protein interactions that have very small interfaces from the PDB, and we try to benchmark like how our Rosetta fold and AlphaFold would behave on PPIs with very large interface, meaning they're stronger and obligate interaction, and PPIs with very small interface. To our surprise, we were able to find that like our Rosetta fold actually perform better than AlphaFold on these transit interaction. Although it's obvious like both tools will work much worse for more transit interaction, but Rosetta fold showed the advantage over AlphaFold. We think it's related to that AlphaFold still was optimized on protein structure. So it's not used to see transition interactions with a very small interface. It's simply by physically they don't like it. However, based on pure on coevolution without thinking about the structure, our cell for PPI seem to be having a unique advantage for transition interaction. So then with this tool, we were able to apply them to do a comprehensive screen of human intactome. So we do have like a de novo screening pipeline, but we also coupled with some experimental guided pipeline where we take like um, protein pairs with existing experimental evidence and try to identify more confident interactions from them. So and as we run our screen, we constantly monitor the performance and check and decide what is the good cutoff scores to use. So this is also another question that I always get asked. So people was wondering like if you use these Russell PPI or AlphaFold to try to infer interactions, then what cutoff you should use. So I would point out if you don't have any previous evidence, yeah, so there should be a curve. I didn't put it in. If you don't have any there's no evidence, then I would only trust a prediction where you get like a interaction probability above 99%. So because they are such a big noise, so you only can trust a very confident interaction. But the good thing is if you do have some previous evidence, like for example, if you take pairs from these databases with some like large scale experimental evidence, then you were able to significantly lower the cutoff and while maintain a high precision. So you can lower the cutoff to about like 0.5, then you were able to get a meaningful accurate prediction by comparing the existing experimental evidence and these AI networks. So and then so we just applied then applying this to the human, and we got 18,000 of predicted interactions, and it's very similar to what we see in the East, that a lot of these interactions they had some experimental evidence. Although experimental evidence, as I introduced you before, is not sufficient to really confidently suggest interaction, but the combination of experimental evidence and our prediction make it sufficient to believe that they are very confident. And a smaller fraction of them was have experimentally determined structures, and about a quarter didn't have um, previous direct evidence for their interaction. But if you study hard, then you were able to find some additional supporting evidence for their interaction. So then again, another important contribution is to provide the 3D structure data for this confident interaction. And these predicted structures just almost tripled the number of human PPS with some high quality structure models. And using these structural models, you can do a lot of functional inference. For example, you can identify the post-translational modification sites or other functional sites or disease mutations on the protein-protein interface. And then the fact that they are on the interface already speak a lot about their potential potential impact. So then very importantly, using this tool, we were able to identify novel interactions that was not observed previously. One important, very good sign of our tool is it seemed to be good at predicting interactions between transmembrane proteins, which was a hard target for experiments. That's why a lot of our novel predictions is related to membrane protein, and in particular GPCRs. So below, I just have a couple of predicted interactions involving GPCR. Some of them, they are upstream molecules or downstream molecules of GPCR. They are also interactions between GPCR and between GPCRs and other transmembrane proteins. So if any of them, if like validated, they could lead to a really exciting biological discoveries. So another thing we can use these our pairwise interactions with screen that can help us to identify new components to known complexes. So here we just showcase a couple of known protein complex, and we were able to identify new components if that component is interacting with at least two known components. So the probably that so then we potentially can add a new component to these complexes. We also can identify protein complexes de novo. So here we showcase a couple of complexes that are dominated by the new members. So then if you dig into all the pairwise data, then you can try to identify new complexes that were missed by experimental studies. So then, since I'm running short on time, I will just skip these slides, and maybe if you guys wonder, then you can ask this question. So then, this is my view of the future of this artificial intelligence. So we think like using all these AI network and a lot of data, we can make a very realistic hypothesis about protein function. And then, but it's more important that we should collaborate with experimental biologists to turn these hypotheses into true validated discoveries. So. And with that, I'd like to thank my lab members and my collaborators that contributed to this project. And yeah, so with that, I'd be happy to take questions.